In Chapter 3, we're going to learn all about the cell in the first part. The human body contains 75 trillion cells. We have 260 different types of cells that are used to build all the tissues that we have and form all the different organs. So imagine all those 75 trillion cells in the middle of your body working all the time. Cells vary greatly in size. We have some very small cells and some very large cells relatively. The smallest is the red blood cell right here. This is, we're going to have a red blood cell. Below that here, we have a white blood cell. Then this very large cell compared to that is the human egg cell. And then this very elongated cell is a smooth muscle cell. The cell is the basic organizational structure of the human body. As I said before, we have 75 trillion cells in the human body. Cell size is measured in what we call micrometers. Micro is 1 × 10⁻⁹. That is very, very small. In order to become all the different types of cells and tissues that we have, cells must differentiate. This is when they specialize and change in order to become what we need. As a result of that differentiation, cells vary in size and shape, which is going to, of course, be based on what function they need, because shape determines function. So I have placed in your unit here this cell size and scale link that you can go and move through it slowly and look at all the difference and compare the different things that they have in there. It's not just cells. They have viruses and bacteria, organelles, and those types of things. And just move through very slowly so you can kind of get a grasp on the scale and size that the human cell actually is. Cells have lots of very different shapes because shape determines function. So cells have those specialized characteristics when they differentiate. For instance, you can look at this nerve cell here on the top here. Look how very different this is from these epithelial cells here, which are part of tissues like your skin. A nerve cell has those long extensions that allow it to transmit those electrical impulses and allow the nervous system and body parts to communicate and go all the way from the brain all the way down to your tippy toes. Epithelial cells are designed to overlay and protect underlying cells. You can see we have some flat layers there. The structure of that really is predicated by the function that is needed of that particular cell. You can look at these contractile proteins, these muscle cells. They're long and stretchy. What do your muscles do? They contract and release. So their function is directly tied to the way that they are made and put together. So this is a composite cell. This means it's kind of a generalized cell here. Not all cells look exactly alike inside. They don't look exactly the same. Some might have more ribosomes. For instance, muscle cells need to make more ATP, so they have more mitochondria. So the different structures that you find in there are going to be based on the needs of the cell. Specialized cells then differ in the number of organelles, which of course is based on their function. We have 3 major parts of the cell. As you can see here, there are many parts, but the major parts are going to be the nucleus, the cytoplasm, and the cell membrane. Those are easily seen under the microscope. Some of the other organelles you have to have special equipment to be able to see. You do want to be able to be sure that you are able to label all the parts of these cells. You will definitely be tested over this in lab and probably also in lecture. So be real thorough, go through your APR. I really highly recommend that you draw these out. Drawing in anatomy is a great way to learn and to concrete that information. The cell membrane is kind of like the outermost limit of a cell. It's got a very active function. We have important metabolic reactions that take place on the membrane, and it contains molecules that allow communication and different interactions. It is selectively permeable, and that means it controls what goes in and out. It is very particular with what comes in the cell and what comes out of the cell. Molecules that are soluble in lipids, which is like oil, pass through very easily, things like oxygen, carbon dioxide, and different steroids. Molecules that are water soluble, however, can't pass through as easily. Remember, we've got this hydrophilic head. Well, let's see. We've got this dual nature here. We've got one part that is water loving and one part that is water fearing. And so that's part of the way that we regulate what goes through there. So molecules that are water soluble don't pass easily through. And those are going to be things like amino acids, which are very large, sugars, proteins, nucleic acids, and then some ions. So they basically have to have a special way to get through. Selective permeability, again, that means controlling exactly what can move in and what moves out of the cell, and even sometimes how many, what the rate is that they're able to go through. The membrane is made up of or constructed of that phospholipid bilayer, meaning there's two of these that face each other. It's made of, has proteins embedded in it, a few carbohydrates. The cell membrane is what we call fluid, and that doesn't mean a liquid. That means it moves and flows. And you're going to watch a video about the cell called The Inner Life of the Cell, and pay attention and look for that. When you see the membrane, you'll see it kind of ebbs and flows and it moves. It also means that there are certain things in the membrane that can move as well. So it's not static. Proteins can move side to side, or they can move across trans in those different directions. All right, so I didn't realize I had to start recording. Cells need to be able to attach to different molecules, but not always form permanent attachments, because sometimes we have cells that we want to be mobile. We don't always want cells to stay put. So think about the fact if you have a wound, you need to get white blood cells to that wound in order to start healing. So cellular adhesion molecules guide these cells that are on the move. Those are going to be those peripheral proteins. We have selectins, and those coat white blood cells and provide traction and binding to help them move along. Integrin guides white blood cells and directs them through capillary walls where they're going to collect on that injury site, and then they're going to be a main component of pus there. Okay, so these are very important. So the cytoplasm, when stained, it appears clear and it has specks in the regular microscope, but it has a very vast and complex network of membranes and organelles. The cytoplasm has protein rods and tubule that form what we call the cytoskeleton. We have the endoplasmic reticulum. This is an organelle. It's a network of interconnected membranes, and they look kind of like flattened sacs that form canals, and their job is to package protein molecules to be secreted. We have the rough ER, which is going to be rough in appearance because it is studded with many ribosomes. Ribosomes here are sites of protein synthesis. That is really important, okay? Smooth endoplasmic reticulum, or smooth ER, does not have ribosomes, so that's why we call it smooth. And this is where lipids are made. Fats are absorbed and broken down. So it's really important. This is a common thing that I see students miss all the time. Rough ER is where we make proteins. Smooth ER, we make fats. Okay, so rough ER has the ribosomes for protein synthesis. Smooth ER does not have ribosomes, so fats, lipids are made there. Okay, be sure to keep that straight. Don't miss that on your test. Ribosomes can be attached to the ER. They can also be floating around in the cytoplasm. So there's also protein synthesis that occurs in the cytoplasm on the ribosomes. So ribosomes are where protein synthesis occur. Okay. The Golgi apparatus is a stack of flattened membrane or membranous sacs, and we call those cisternae. Kind of like a cistern. A cistern holds things and it's open on the inside, right? Their job is to refine, package, and deliver proteins. They have vesicles in the membrane sacs that function for storage. All right, mitochondria. What are mitochondria? Do you remember what mitochondria are from general biology or from perhaps high school biology? Well, most people say they're the powerhouse of the cell. Well, let me ask you, what does that mean? What does it mean to be the powerhouse of the cell? So if I ask you that question on the test, don't tell me they're just the powerhouse of the cell. I want to know what they do. They're membrane-bound organelles. They have DNA with the information, the instructions to make proteins and RNA. This is where ATP is made. That's why we call them the powerhouse of the cell, okay? But they make ATP as their main function. We have an inner and an outer membrane partitioned into what we call cristae. You can see that here. So we've got an outer membrane, and then we have this inner membrane, okay? And then they are going to fold, and we call this enfolding cristae. And this is where we have surface reactions occur. Most important one that occurs there is cellular respiration. And what is the goal of cellular respiration? Well, I hope you thought about that word I said, ATP. Okay, ATP is the energy currency of the cell. And so that is the main function, is to generate or make ATP through cellular respiration. So this diagram, and if you'll look at the diagrams in your textbook and read them and try to understand them and read the little captions that come with them, you'll find that they're really very, very, very useful. Be sure that you understand these. Take the time to go through and really look at these. So if we start over here at number 1, well, first off, this is going to be milk secretion. And so this is going to show us how organelles interact together in order to synthesize or make, transport, store, and then export different biochemicals. So we're looking at milk production here. So lipids, which are fats, milk contain fats, those are going to be made in the smooth ER, okay? And then we're going to have milk protein being made. That's going to have a gene here in the nucleus that are going to be transcribed into mRNA. So the instructions to make the protein are going to be transcribed here in the nucleus, okay? Then the mRNA has to actually leave the nucleus and then go where? Well, it's got to go where we make proteins. So most proteins are synthesized on ribosomes that are associated with membranes of the rough ER, okay? And so those are going to use amino acids that we find in the cytosol. What else does milk have in it? Well, we've got our protein, we've got our fat, and milk has lactose, it has sugar. So sugars are going to be made in the smooth endoplasmic reticulum and the Golgi apparatus. And they can also be attached to proteins, or they can be secreted in vesicles. So we can do different things with those. Proteins are going to be secreted from vesicles, and those are going to actually bud off of the Golgi apparatus. And then fat droplets are going to be able to pick up a layer of lipids from the cell membrane as they are leaving the cell. And so all of the ingredients, it's like a little factory, are made in different places, and then they're all put together and shipped out. So it's really quite fascinating that our cell does all of this in every 75 trillion cells that we have, in all of them. We have lots of different organelles in the cell. You need to know what they are and what their function is. That is very important. Lysosomes here, these act kind of like the garbage disposal of the cell. They contain an enzyme that is going to take worn out cell parts and dismantle them and pick up debris, okay? These can be kind of hard to identify in a cell because the shape varies some. Then we have the centrosome, and you can see that picture here at the bottom. It is located usually near the nucleus. It participates in things like cell division. It also helps form parts of cilia and flagella, okay? So most importantly is the participation during cell division. It actually helps distribute the chromosomes there. Next we have peroxisomes, and these are kind of like lysosomes in size and shape. They look similar. These are found most abundantly in cells in the liver and the kidney. This contains what we call catalase. And I want you to remember this because you're going to see this again. Catalase that helps to break down or decompose hydrogen peroxide. So it's going to break down H2O2, okay? Because that is a byproduct of some reactions in our body, and we're lucky that we have catalase and we're able to break down H2O2 because it would be toxic otherwise. We can get toxic forms of oxygen, which is something like we normally hear of is called like a free radical. So we have next cilia. These are short hair-like projections. You can see those up here on the top of this cell, and they propel substances on the cell surface. This is going to be things like moving mucus out of your body, which is a very important function for health. Okay, it's to help keep you from getting sick. It's what we call one of the first lines of defense with immunity. Then we also have flagellum, and I want you to remember not all cells have cilia, not all cells have flagellum. Okay, sperm is the picture here that has flagellum that we are most familiar with. They have a long tail-like projection. And what is the purpose of that tail there? Basically, it's a tail, right? What is the purpose of that? Well, to help the sperm swim, right? To help it move around. So that's pretty important. But remember, that's not on all cells. We talked about the cytoskeleton and some of the components of that. Let me write that word on there for you. Cytoskeleton. And what we do in class usually is I have you draw a cell, label the parts, and then give me the function. I haven't quite decided yet how we're going to cover that, but it's really important that you be able to do that because then you're able to remember it very well. And we're going to be looking at these different parts of the cell, and it's going to come back around in microbiology, which most of you are going to need to take as well. So you might as well do the work and go ahead and learn it now, all right? And you really should already kind of have the basics of this. This should be a refresher. So we have microfilaments and microtubules. These are thin rods and little tubes, and they act mostly in support. They also allow for movement of organelles through the cell. So again, when you watch that video on the inner life of the cell, I want you to watch for specifically how those microfilaments and microtubules are used. And you'll notice in the video that they're not static. They're not in the same place all the time. They're actually formed, and then they break apart, and then they're remade. And so you'll see them coming together in the video, and you'll know what I mean while I'm doing this when you see the video. It's really a cool video. As needed. It's on an as needed basis. So that's really pretty cool that our cell is constantly evolving and changing in that manner. It's not static. It's always changing. You can see here in the picture that we have the microfilaments have a little bit different look. They're a little thinner. The microtubules are a little bit larger because they are tube shaped. Be sure when you're looking at these on APR that you pay attention to which is which, because that's another commonly missed question, and that would be on the lab exam. Okay, and so this isn't lab, but I'm thinking about it now, so I want to make sure I point it out to you. So pay attention on APR which one of those are which, because they look very, very similar. And there are some things that APR is just dynamite for illustrating, and some things that are a little harder to tell. It's easier for me to look at this diagram and tell the difference between a microfilament and a microtubule, a little harder on APR. So when you recognize that and things look very similar, take a note to yourself and say, okay, I need to be able to identify which of these is which and look for those differences in those things. So the cell nucleus is the control center of the cell. It's kind of like the vault where you keep all the really, really important information and you lock things up that are really important. So we have a nuclear envelope around the nucleus. It's made of a double membrane. It contains these little nuclear pores. You can see things here that allow substances in and out. Okay, it's a small, dense body. The nucleolus, it's a small, dense body of RNA. You can see here this whole thing is the nucleus, and this darker portion is the nucleolus. So it contains RNA and protein. It does not have a separate membrane. It contains nuclear pores for ribosomes, which are produced there. So the ribosomes are made in the nucleolus, and then they're shipped out through those nuclear pores into the rest of the cell. Chromatin is going to contain the 46, that's the number for humans, 46 chromosomes, which contain our DNA. And we have chromatin fibers that coil tightly together, making the individual chromatin fibers visible with light microscopy. And of course, that depends on which part of the cell cycle you're in. And we'll look briefly at the cell cycle, and I think this chapter, the last PowerPoint, which I believe is section 3. I think that's where cell cycle is. And so this stores the genes in order to make proteins, and it stores the instructions. Okay, very important.